Thank you. Thank you, Marty. We started Thrive Bioscience about 6 years ago in Boston because we found an incredibly important area of life sciences that was almost completely ignored, which is cell culture. So next slide, please. So Thrive makes instruments and software for researchers to basically be able to study cells better. My own background, by the way, I've started 11 companies. I have 4 multi-billion-dollar exits. My first 3 companies were in software, and the rest were in life sciences. A comparable company is Cytic, where we automated cytology labs instead of cell culture labs. And we sold that company for $6 billion. 90% of all pap smears are now read with those instruments. So we have a team of very experienced people. My co-founder is really quite incredible. He invented the sequencers that Illumina uses, which are the most widely used in genomics. And our chairman was president of Thermo Fisher, the largest company in this area. We have significant collaborations with a big pharmaceutical company, Otsuka, in Japan, and with 11 research institutions, the Broad Institute, Harvard Stem Cell Institute, University of Texas Medical Branch, Stanford Stem Cell Core, and others. There's very limited competition in this area, and as a result, we've been able to put together a really comprehensive IP portfolio. We have 74 patent applications, of which slide says 24. I just heard we have 25 issued. So we expect sales of about 5 million this coming year. The year after 19 million, and in 2026 over 150 million. That's without an acquisition, which we're hoping to close shortly. So we're raising $8 million under a convertible note that converts into the Series B at a 20% discount. We've raised 4 million of that. We're over. This is a convertible into the Series B.
And what's the interest rate?
At a 20% discount and a 5% interest rate, which accrues. You're not in it for the coupon.
No.
We started with 1.5 million, so we're quite oversubscribed. The reason that we're raising
8, and what's left?
I'm raising 8, and we have 4 left because we just found out a company that we know very well, which is a supplier, is for sale. And
So you got 4 left.
Right. So we hope to acquire that company. It will do about $7 million next year and is profitable. Next slide, please. So we have this saying, and the saying is good science needs good cells, and that's the title of this talk. So I want to tell you about cell culture. Cell culture is an enormous market, over $18 billion per year. And it's basically key to the biomedical research. Almost all research is done in cells. And it's conducted manually, just like it was 65 years ago. Hours are spent looking at a microscope at cells, and then the cells are put back in the incubator. And guess what? All the data is lost. And because of the fact that it's manual, these cells vary by experiment, by lab, over time. And so we have data that's highly variable and non-comparable. And then we take these cells we know very little about. In fact, I would say Uber knows more about us than we know about ourselves. And we put them into incredibly expensive instruments that we spend billions of dollars a year on. But the variability of the cells swamps the outcome. And so this is part of what's known as the reproducibility crisis. Next slide, please, Marty. So I just want to give a little review of what cell culture is. And basically, we're recreating in a laboratory.
What's under that mask? On the title?
So artificially recreating cells, environments to grow cells in the lab. And we do a terrible job at it. Mislabeling rates are 11% to 34%. This is extraordinary, but it's common in an unautomated process.
That's bigger than human error, like the average human error.
Because it's accumulated over 65 years. And so there's a 34% chance that a researcher doing drug discovery, which is our largest market, is studying what he thinks are liver cells, but they're actually pancreatic cells. Those statistics come from National Institutes of Health. Next slide, please. So we really fail at doing cell culture, even though it is the basis for much of research. And in fact, Nature magazine talked about it as a crisis rocking research. And the first real bombshell came when Amgen did a study with 100 scientists over 10 years to try to replicate 53 landmark studies in oncology. And they could not replicate 90%. And 50% approximately of the cause of not being able to reproduce was traced to cell culture. So cell culture basically is slowing cures and placing patients at risk because non-reproducible research, it gets cited on average. Non-reproducible research is cited within 2 years, an average of 2,000 times. So we're accumulating this knowledge, which is really not knowledge. And we're also putting patients at risk. In a 7-year period, over 125,000 patients were in clinical trials where the research was later found to be not reproducible.
So now, are you talking
Next slide,
strictly about
please.
cell culture in terms of a production?
Marty, next slide.
A production methodology for proteins and peptides and what have you? Or are you looking at cell therapy, cell stores as well?
Next slide, Marty. So very good question. So cell culture does all those things. And we're starting with the research market because it's
not refreshing the screen.
There we go.
It's taking a while. Sorry about that.
Sure, no problem. So we're starting in the research market where those things are done in a small scale.
Right.
But an even bigger opportunity for us in 3 or 4 years is manufacturing or bioprocessing. And we're getting pulled there more quickly than we thought. It's a different market. It's a fabulous market, high margins, long sales cycle though. But once you're in, you're written into an FDA good manufacturing process, you're there for 10 years. But they always want validated instruments. So it's not a great place to start. Research is a great place to start. People are open to new products.
But are your equipments there for sort of analytical purposes or for part of the manufacturing line?
So it's being used for really breakthrough insights and documentation. So we are creating these enormous databases of cells for the first time in live cell biology. There are, we have over 12 customers, several that are producing, but at a small scale. But nevertheless, they are trying to control their processes, which are almost impossible currently because you don't have consistent information.
So in other words, you would be selling to somebody like ATCC.
Yes. So ATCC would be in the middle between research and production.
Right.
But they actually have compared our instrument to other instruments and said that our analytics was the best that they could find. So we expect to have a good number of sales there, dozens and dozens. But they are really a bioprocessing manufacturing. But they do some R&D to optimize the processes, and we play there. So what's really unusual about our instrument compared to any other instrument is we're the only instrument that can actually find and track a single cell or a colony over time. This has been the holy grail that's been unachieved. So that means you can actually study what goes on in that cell and get back to that cell over weeks, over months, because our customers take data every 3 hours, every 4 hours. They'll image our plates as part of creating their database. And we capture up to 60 focal planes. We can actually go higher, but we had to pick a number, 2.5 microns to 50 microns apart, so that we're actually creating 3D type images because biology is not flat. And one of the fastest growing areas in cell culture is actually tissue culture because it's a model between cells on the bottom of a plate and animals. So a big gulf in between there. And so there's a lot of research in tissues.
What is do you use EM for your imaging capture?
So we use very basic microscopy.
Just photo microscopy?
Yeah, bright field and phase contrast. And
So Tom, you have about 10 minutes left. So
Okay.
we either want to table the questions and get to the end,
Sure.
or I
Sorry,
need just
sorry.
to figure out which way you're doing it.
If we get the slides back up, that'd be great. So while we're getting the slides, we have a family of products. We have two on the market. One is a benchtop imager, takes a single plate of cells at a time, and what we call the Cell Assist 50, which automatically images cells, 50 plates, and it can do it every 2, 3, or 4 hours. So the applications are some of the most important applications. Next slide, I think, right? And I did the next one after that.
Next one.
Yeah,
I'm on
and
slide.
the one after that too. I think I went through them.
Collaborative cell assist, I'm on.
Could you go to the
Sorry.
next? So I want to show some images here.
The regular cells.
If you can go back, maybe one more, go back.
Okay. Okay.
You go back. Sorry, I think you went forward. And just one more back. I'm not, just so I know what's there. So this is an example of what data can do. Remember, I told you that there's actually 11% to 34% mislabeling. And here's an example from the Broad Institute of Harvard and MIT, where for the first time, they're able to catch the fact that they were mislabeling cells. Of course, everybody thinks they don't do it, but the power of data can really increase the quality. Next slide, please. So I wanted to show you some images of what people are using our plate for. This is incredible. So the person, I think we missed the slide, but a chairman of the biochemistry department at University of Texas Medical Branch said that Thrive will change the world. And it was because of these images. So they're the leading center for infectious disease research in the United States. Galveston National Laboratory is where Andromeda bugs go. This lab was credited by Pfizer for being one of the principal reasons that they got their vaccine to market first. And they're using our instrument. For the first time, they're able to characterize what a virus does to cells. The current standard is that you put an overlay of the virus on cells, you wait 7 days, you fix it, stain it, and take a look at it. So you have data at one point in time, and you have to touch the cells again in order to stain them. And so the challenge that they used our instrument for was to not have to stain it, so that they didn't have to handle coronavirus again, and so they could continue to follow the cells. We achieved that easily. They then said, we would like to understand how the virus forms, because currently no instrument can do what you see there, which is watch a plaque forming over 7 days. And actually, as a result of these images, they discovered that they had a variant in their cell culture plate, which was confounding their data. They were screening for drugs. So this is something that has never been able to be done before. Next slide. So this is an example of tracking a single cell over time. A little bit hard to see from, unless you're close up, but there's a single cell all the way on the left. It doubles and continues, and it actually turns into 2 stem cell colonies. And the gold standard actually in growing stem cells for regenerative medicine and research is that you have a colony that's homogeneous genomically, but nobody can tell that until the cell assist. Next slide. So I mentioned tissues and organoids. This is an example of actually a stem cell derived organoid that the Broad Institute was imaging, and they said that we are the best instrument for imaging tissues. Next slide.
About 5 minutes.
So I gave you my background a little bit about Alan, who after 25 years in genomics, producing huge amounts of data, Illumina 90% market share, and he built those sequencers. He and I want to do that for cell biology. We have 17 people at the company. Our chairman was president of Thermo Fisher, and another one of our outside board members, Brock Reeve, head of the Harvard Stem Cell Institute. Morning. So on the financing side, we've raised $29 million to date. You can see valuations have risen nicely from post money of 10 million to 23 million to 40 million to 65 million. We are going to target completing our Series B for $10 million mid next year. And the note that converts into that for 8 million, we hope to complete this month, so we can acquire that nice little profitable company that's one of our suppliers and is focused on environmental controls in cell culture. So it's really a great fit. Our largest shareholders, the Estee Lauder family, they have invested in 2 of my previous companies, HNC Software and Cytic. We also have the Milstein family, New York Real Estate, the Parker Brothers family, Monopoly game. I actually am spending Monopoly money, I guess, and I always wanted to do that as a kid.
Make it.
And we have 7 angel groups, including Life Science Angels, Sidecar Angels, New York Angels, a lot of industry veterans, former CEOs of Novartis, Cytic, Patheon, Johns Manville, and actually our IP attorneys think so highly of our. It's not because we couldn't pay our bills; they voluntarily.
Is that Life Science Angels out of New Jersey?
Sorry?
Is that Life Science Angels out of New Jersey?
No, California.
Okay.
Next slide.
And Lauder, is that the Lauder group in California or in New York?
So both have invested, both Lauder
Gary.
partners, Gary Lauder
Okay,
in Palo Alto area, and the New York family office from several funds has also invested.
Interesting. Okay, so great.
How much has the founders contributed?
I've put in $2 million of the 29.4, and Alan's put in, my co-founder, $500,000. Next slide, please. So in terms of returns, obviously these are hard to predict, but I expect this to be my largest exit because it's larger than the cytology market. It's not FDA regulated, and it has many segments. It's applicable to almost all of life sciences. So we expect a 20x return based on our projections and comparables. Multiples for publicly traded companies in M&A for our type of company with under 300 million in revenue is 10 times to 35 times trailing revenue. But we're most likely to be acquired. We fit well within 15 to 20 companies such as Nikon, Fujifilm, Thermo Fisher, Lonza, PerkinElmer. And we've been contacted by all of those, actually. And the public company comps are really excellent as well, about 35. They're trading at about 35 times trailing.
To what
revenues.
extent is all of this IP?
Enormous, everything. You have to buy our instruments or buy us. We have 74 patent applications in all areas of software, machine learning, use of blockchain, some new types of microscopy. We actually modify, even though we use phase contrast and bright field, we do it in a different way and have very advanced algorithms for focusing and things like that. So I'd say of the 11 companies I started, I learned the importance of IP when you're at the stage of selling. And actually, current companies on that list are violating our IP, but we're not about to get into fights about that until we've filed a lot more. And that may be the last slide at 19 minutes and 30 seconds.
Great. Why don't you, any more questions or questions from the audience? Let me just take this down.
Question.
Sure.
Largely, your investor base seems to have been angels, high net worth, family offices. At what point do you think you'd be ready for an institution or an institutional or institution like venture fund who's involved in your space?
Really excellent question. So the venture funds typically do not invest in our segment, life science tools, until they're revenue generating. And a lot of that has to do with the fact that instruments and biology have created a lot of problems for a lot of companies. So in fact, this coming round, we expect to be entirely a venture round.
You think you're ready for it now with the acquisition you're making?
I think we're ready without the acquisition. It'll be
Right.
VC backed, but having that acquisition moves us faster to an exit, either a public offering or to be acquired. We see that company's products in every single lab that we visit.
You have big dreams, and I think you're going to reach them.
Well, thank you very much. It's that vision that keeps you going when the going gets tough.